Okay, I'm going to explain today what translation is all about here. And before we begin, I thought we'd just kind of review where we are. So what you're looking at right now is the basic paradigm of molecular biology. Paradigm, remember, means just the way things work. So if we look at our information, it's stored as DNA. And this would be DNA just starting at the beginning here. And then DNA, Watson-Crick discovered, can be reproduced in mitosis, and that's what we call replication. So you can see here that DNA can just be made into DNA again, and we saw that that happens by the two strands separating, and then a DNA polymerase came in, matched up the bases, G to C and A to T, and made new DNA. So that brings us right here at DNA. This takes place in the nucleus. Now there's another process which takes place in the nucleus, and that is DNA is transcribed into RNA. So DNA can be made into RNA, and there's three types of RNA that can happen to. This also takes place in the nucleus. And the reason why this is happening is that the information which is stored in DNA is going to be used to direct everything going on in your cells, but we don't really want the DNA to leave the nucleus because it's very valuable, so we kind of keep it in that locked box there. So RNA then is made in the nucleus, but then leaves the nucleus, goes through the nuclear membrane, goes out into the cytoplasm. That's where we left off last time. Now what we're going to talk about today is, so what's the purpose of that RNA? Well, the purpose of that RNA is to produce all the things that your cell needs and your body needs, and to make you into you. And that means it has to direct all the proteins it needs. So proteins that make your eyes the color they are, and makes your skin color the color it is, and your height the height it is, and so on. We call that process there translation. So this is translation here. RNA made into proteins. So let's look at it again. DNA can be replicated into DNA, can be transcribed into RNA, and then translated into proteins. Let's go to the next screen now. Now, just to review transcription, which we talked about last time, there's actually three types of RNA that the DNA can be transcribed into. And the three types are mRNA, which stands for messenger, rRNA, ribosomal, tRNA, transfer. And those three types all have the matching pattern of G hooks up with C, but this time A hooks up with U. And the reason is there's no T, there's no thymine, and this guy U, and we can spell it out if you want to, U is going to be a new type of base, and it is going to be called uracil. So there's no thymine there, just uracil, and that's one of the ways that we know that this is RNA and not DNA. Let's go to the next screen now. Okay, now so you have a long piece of RNA. Let's say you have a long piece of mRNA. And the way it works is that every three bases in that RNA is going to be called a codon, C-O-D-O-N. And what happens is that it's a long code, which is going to be translated into protein. And the way it works is that another type of RNA, which is going to come along, which is going to be rRNA, and that's going to come along and read it. So let's suppose now we have an rRNA that's going to read this piece of mRNA. So it comes along here and reads that portion right there. And the first codon it reads then is CGA. It comes along, it reads it, it says, okay, and then it goes to the next one. And then it sees an AUG, and it goes to the next one, CCA, and to the next one. So it's like glasses, it's like a reading frame here. Then GAU, and then it goes to the next one, and CCU. So RNA is kind of like a reader, and it's reading every three bases, and every three bases will be called a codon. Okay? Let's go to the next screen. Now, at the same time it's doing that, another type of RNA comes in, which is the tRNA. So you see how all three types of RNA, mRNA, tRNA, and rRNA, will all be involved here. So what does a tRNA do? Well, the tRNA kind of matches the mRNA. It has every three bases on it, but its three bases aren't called a codon, they're called an anticodon. And those anticodons are going to be complementary matches with the codons. Remember how it works? C matches up with G and A will match up with U. Let's take a look at this for a second. Okay, let's suppose that we have this strand of mRNA up there. So that is CGAAUG, CCAGAUCCC. Okay, the rRNA reads it every 3, so it reads these first 3, CGA, and it would bring in then a tRNA which has an anticodon that matches it. And that'd be this guy right here, yeah? Then it reads the next one, AUG. Who matches with that? Well, it's going to be UAC. That's this guy over here. Okay? Then it comes in and reads the next one, CCA, and it's going to come in here and match up GGU. That's this guy right here. Then who do we have? We got GAU, and that's going to be CUA. The last one is GGG, and the anticodon, the tRNA, would be GGG. So you see how the idea of complementary bases keeps working here, only this time it's mRNA up here, and we have tRNA down here. Let's go to the next screen. Okay, what's the purpose of these tRNAs coming in? Well, each one of these tRNAs carries an amino acid. Let's see, I misspelled that. Let's put an R in there. Now, there are 20 different amino acids, and you have to eat all 20 of them every day to stay healthy. And to make things easier, they're abbreviated by 3 letters. So, for example, an amino acid called methionine would be abbreviated as MET. Now, here's how it works. As the mRNA is being read, 3 bases at a time, 1 codon, right? Amino acids are brought in by those tRNAs. They're hooked together to form a protein. Remember, amino acids make proteins. This whole process is called translation. So let's take a look now at everything all put together. Here was our mRNA strip again. Here are the tRNAs with the anticodons on them, but this time you can see that each one is hooked up to a 3-letter amino acid. So let's put these guys in. The first guy is CGA. That should match up with GCU. So that would be this guy here. And it's going to go ahead and bring in an amino acid called ALA or alanine. The next codon is AUG. That will match up with UAC. That brings in tyrosine. Then we got CCA. That would match up with GGU. Brings in cysteine. Then you got GAU, which matches up with CUA, which brings in an amino acid called leucine. Then you got CCC, which matches up with GGG. That brings up glycine. And what's going to happen then is that all of these guys are actually going to be hooked together with bonds like that, which then forms a big protein whose sequence is alanine, tyronine, cysteine, leucine, and glycine. And this goes on and on for quite a while. Now, the trick is, though, is how long does it go on? What controls this translation? Well, mRNA is very long, but only a small part of it is actually translated into a protein. How does that work? Every protein always starts at the same